Okay, in this uh, unit, we're going to be talking about DNA technology. So, some of the things we're going to be talking about are like regular DNA fingerprinting versus like a regular fingerprint. If you get arrested, they take your fingerprints on your fingers. Well, there's something new out there now called DNA fingerprinting, where we look at the DNA of a person. You ever heard of cloning? Cloning is like a miniature mini me, identical to you in every way except in age, because it won't be created at the same time that you were born. And then GMOs, or genetically modified organisms, which you're eating now, whether you realize it or not. So those are things we're going to be talking about. And the first thing, you're on the first page, top left, next to the picture of the dog where it says selective breeding. You're writing everything that's in red. Man has been selectively breeding since 3000 BC. The pharaohs created what's called the pharaoh cat, which they mated cats together to get the features that they wanted until that became a breed of cat after a while. We've done the same thing with dogs. This is a Great Dane. So you breed all the largest dogs together to get your Great Dane breed. Same thing with like a Chihuahua. You're breeding all the small dogs together until you get a breed that looks like the modern day Chihuahua. And old Bessie the cow here, she also has been genetically engineered to produce more milk. These cows have been hooked up to automatic milking machines. People don't have to do that anymore. They just hook them up to the machines and the machine will take care of it because they produce so much milk. Now, the root word of genetic engineering. The root word of genetic is gene, so that's DNA. Engineer means to build or to design. So, in your term, genetic engineering, underneath the dog in your notes, it's when we choose to alter an organism's DNA. But when we alter that DNA, we now call it a transgenic organism. Trans meaning across, genes. So transgenic organisms have genes from other species mixed in with them. So we're altering the gene in some way. So genetic engineering is just a generic term. The example that we are going to be looking at are transgenic organisms because we literally put DNA from one organism into another. Now, if we try to remove a genetic disease from a person so that they would automatically be healthy again, that would just be called genetic engineering because we would be changing the DNA of that person so they would no longer have the disease. We're not quite there yet. We're still trying to figure that out. So here's some transgenic organisms. And in your notes, you have a picture of a corn plant. We make in Pitt County, we make BT corn. BT corn is pesticide resistant. You're copying what's in yellow. This corn plant, in a gene in its leaves, has a poison gene from an insect. So they took an insect that produces a poison. They took the DNA for that gene out of the insect, and they put it in the corn plant. So it's all on the leaves. When the corn grows, that gene is used and makes the protein that's embedded in all the leaves of the corn plant. So when a leaf-eating happy grasshopper comes along and thinking, great, I've got lunch for the rest of my life, starts eating it, it dies. Now, when we eat food from transgenic organisms, which is what BT corn is, we call that a GMO, genetically modified organism. Now, it's important to note that humans don't directly eat BT corn. BT corn is what we call feed corn. That's what you give your horses and your pigs and your cows. Humans can't eat it. It's that really hard stuff. But we do make this in Pitt County, and it is an example of a transgenic organism. Some other examples which you've eaten: frost-resistant strawberries. which, as the name sounds, it gets cold outside, the plant freezes. But frost-resistant strawberries, the plant doesn't freeze because circulating in the system of the plant is a protein that makes it, that keeps the water inside the plant from freezing. We actually get that from an Arctic fish. They have a gene that they make that goes into their bloodstream to keep the blood from freezing because they live at the North Pole. So this gene, we take the gene out, not the taste of the fish, just the gene that actually makes the protein. And then as the strawberry plant lives every day, it makes that protein. And then if those strawberries, if it gets cold enough to freeze, the strawberry plants don't freeze and the farmer does not lose its crop. You need to write down what's in red here. The grapple, here's a picture of them, are apples with grape DNA added. And you eat the apple and you get the grape as the aftertaste. But it looks like an apple, smells like an apple, but you can tell there's something else in there. I do try to go around town trying to find some of these to get, but I have not been able to get them for the past few years. But if I can find them, I bring them in and I let y'all eat some. We also add medicine to foods, and corn is huge. This is white corn here. The middle one is yellow corn. Those are the corns that we eat. Okay? This corn we grow in the United States, but we do not eat it here. We ship this overseas, and yes, it's orange. That's because we've added the gene called beta-carotene to the corn. So that when the corn makes the seeds, it stores a lot of beta-carotene in the corn kernel. And that's what you see here. Each little one of these corn kernels is a baby corn plant. We ship the corn overseas for people to eat. Beta-carotene is turned into 2 molecules of vitamin A. And that vitamin A keeps people from needing glasses. So it helps people in countries that are not wealthy enough to afford glasses. They can eat the corn, and that gives them a little bit of an edge there. We also can put drugs and other things in corn. We put an awful lot in corn. All right, now, so how did we do this, make these transgenic organisms anyway? Well, first thing, we used prokaryotic cells in the beginning because they do not have a nucleus. Their DNA is easily altered. Even high schoolers can do this. In my AP biology class, they altered the DNA of a bacteria as one of their labs. So it's very easy to do. Bacteria also have plasmids, which are extracircular pieces of DNA, and they give bacteria little kind of extra genes and things that they can do that not all bacteria can do. What we do is we add the genes that we want the bacteria to produce to the plasmids. And that allows the bacteria to make proteins for man. The example here, the first one we did was insulin. And I mentioned this many times before. Many humans, we don't use pig insulin to give to diabetics anymore. You can actually take real human insulin. We get the bacteria to make it for us. The second thing we had bacteria do was to make growth hormone. Growth hormone, there's about 16 different kinds of dwarfism where people don't grow to normal height. So some of those types of dwarfism, all they need is the correct growth hormone to be injected into their body and they'll grow to the normal size. So if we have bacteria make the growth hormone protein, all we got to do is put it in the needle and stick it into the arm of a dwarf and they grow to normal size. And that's a good thing because dwarfs have normal size organs in bodies that are less than 4 feet tall. So it's difficult for them, for those organs to function, and they do have a shortened lifespan. Now, you've got this picture in your notes. So these little pieces right here, that's a plasmid. This is a plasmid. That's a plasmid. So not all bacteria have them, but bacteria that don't will usually bacteria will take up as many plasmids as they can because that gives them little things that they might be able to use to survive our like penicillin resistance or something like that. When you insert a human gene into a plasmid, that plasmid is called recombined because we've taken the DNA apart and we reordered it in a new way. And so every day that bacteria will make that recombined gene, whatever that is, insulin, growth hormone, whatever gene we put in there, they're going to make it. And of course, if you don't remember, diabetics use insulin to lower their blood sugar. Now, just a reminder, here's the insulin. It lands on like a lock mechanism that opens a membrane protein that allows glucose to travel down its concentration gradient. That's from a high layer of concentration to a low level of concentration, which would be inside the cell. So do you guys remember which type of transport this is? It's passive, so it's not active transport. Is a movement of molecules from high concentration to low concentration using a membrane protein. Is that diffusion or facilitated diffusion? Okay, if you said facilitated diffusion, you would be right. Okay, now let's go up to the top of the right side on that first page. Gel electrophoresis. Okay, it separates using DNA by their size. So what we do is we take DNA, like from a DNA at a crime scene. We use a restriction enzyme. You guys remember, like, digestive enzymes break down food? Restriction enzymes, well, they break down DNA. They cut the DNA into little tiny pieces. Then we take those pieces, which are in this little needle here, this pipette, and then we put them into a gel. This is, if you've ever, cooked with something called gelatin, that's unflavored Jell-O. That's exactly how this thing is made. Hot water, some of the powder, and you have gel. You have a gel for gel electrophoresis. Once you put the DNA in, which is colored purple here, you can actually hook it up to electricity, and DNA is negative. So we load the DNA at the positive end, like on a battery, that's the red side or the plus side. And then we plug it into the power supply, and the DNA will leave the positive side of the gel and will move through the gel towards the negative side. And when you turn the gel off, what has happened is the DNA has separated out into these are called lanes. This is the first lane, what we call EV stands for evidence. Then you have lane number 1, lane number 2, and lane number 3. These numbers here are your suspects, who you think committed the crime. When you turn the electricity on, the long pieces, you need to write this down, the long pieces don't move very far. And these pieces right here, they're about 20,000 nucleotides long. So remember, a nucleotide is a sugar, a phosphate, and a nitrogen base. And there's about 20,000 plus about at this end. The shorter pieces are lighter and smaller, so they travel further in the gel. So the short pieces end up at the bottom. And this piece down here would be probably about 600 base pairs long. So you're like 20,000 plus up here and 500 or so down here. And so what you get is you get these each lane going down would be a DNA fingerprint. The evidence would be DNA found of the person who committed the crime. 1, 2, and 3 are your suspects. The evidence lines, every single DNA band in the evidence lane must be in the exact location and the same thickness to be a match. So which one of these suspects, 1, 2, or 3, identically matches the EV? If you said number 2, you're right. Write it down right here in your line. So if this were a real crime scene, this was real DNA, suspect number 2 would be called in for questioning because their DNA is at the scene in some form or some way. Now, DNA cannot be the only evidence they have. They still have, cops still have to have the 3 M's: motive, means, MMO, opportunity. Motive, means, and opportunity. Sorry, MMO. All of that still comes into play. So let's go down to this. All right, so here is another kind of gel. This is what sometimes people call the Who's Your Daddy gel. So here's a mama, and here's her baby. We know from meiosis we are 50% of each of our parents. So there's 4 lines in the baby. That means 2 lines come from mom, 2 lines come from dad. So we have to identify the lines the mama and the child have in common. So mama's got this line, and mama's got this line. So I'll call that, this is line 2 and line 4 of the baby. Mama's got this line and this line. That means biological daddy must have line 1, which person 1 has, person 2 does not. And then the daddy must also have this line, line 3 in the baby, and that would be this person. So daddy number 1, write the number down. Daddy number 1 is the daddy. He's the one that's going to be paying child support. Daddy number 2 does not have DNA that matches the baby, so not him. Now, real gels that identify a father of a child, they don't use just 2 lines like we did here. It's required to be a 14-line match or more. So we want to make sure that we don't, you don't want to have the baby's daddy be the brother of the actual father of the baby. So you want to make sure that you've got the right person. So it's got to be a 14-line match, not a 2-line match like we did here. But it's the same principles. Okay. So that concludes these